Successfully recording. Okay, so thanks everyone for joining us. Alessandro and I are very excited to kick off the first seminar of our seminar series on the mathematical laws and morphology and biomechanics. So this is a seminar series that's part of a broader initiative that we've emailed many of you about over the last few weeks, spamming your email inboxes. It's sort of an effort that we have started in trying to bring together students and researchers from as many different backgrounds and perspectives as possible to sort of learn and teach and diffuse knowledge about the use of quantitative methods coming from mathematics and statistics and machine learning and data science in order to understand questions related to the biological laws that determine morphology and biomechanics, organismal biology in general, and how those different fields connect. So I will be putting a link to the program website in case any of you have not had a chance to take a look at that. But we have some working groups that we're going to be starting as well in addition to the seminar series. And we have a great lineup of seminar speakers coming up. So without further ado, we would like to introduce Professor Alain Goriely from the University of Oxford for the first seminar of our seminar series. He is a fellow of St. Catherine's College and a member of the Mathematical Institute. He's also the director of the Oxford Centre for Industrial and Applied Mathematics and the director of the International Brain and Mechanics Lab. His work in applied mathematics shows the breadth of his research interests and contributions. He's generally interested in the study and development of mathematical models for applied sciences, and his work includes the mechanics of biological growth, the modeling of the brain, theoretical foundations of mechanics, dynamics of curves, knots, and rods, modeling cancer, and the list goes on. It is a really great honor to have Professor Goriely speak in this seminar series, and we cannot think of a better way to kick off this initiative. So without further ado, we'd like to hand over the floor to Professor Goriely, or in this case, I guess we're handing over the screen to him, and he's going to be speaking about 10 ways to model growth.
Thank you very much, Kathleen and Alessandro, for the invitation. It looks like you have a wonderful program. Let's see if that sharing. Okay, my sharing didn't work, so let me focus. Looks like it's working now. So just okay. I note from you that it's working. Okay, very good. So thank you again very much. It's great. It looks like you have a great program, and thank you for the chance to talk. Just a few exchange with Alessandro. He told me he'd be interested in having some kind of review about the idea to model growth. I guess it goes well with your idea of trying to isolate laws for morphogenesis or law for biological systems. What I'll show you is that growth is so general and come in so many different guises that there is no single laws, but there are different ways to approach the problem and different framework in which we can understand it. More interestingly, if you're interested in geometry, the mathematical aspect is that the problem of growth itself forces us to further develop mathematical ideas that are not the common one that are found in, let's say, a field theory of physics. The idea that a space like a biological entity can grow requires some new thinking. And so not only we can apply mathematical idea to understand growth, but growth itself induces some new mathematical idea, but that's really going to come to the end. So it's really the product of a long journey, a long reflection that started maybe 20 years ago, something like that, or maybe even earlier than that. Essentially, when I was in Arizona, in Tucson, Arizona, I developed some of these early ideas with Michael Tabor. And then after that in Paris with Martin Benamar, then with Derek Moulton, who was at the time in Arizona and then moved to Oxford. He's now professor in Oxford. And over the years, I've collaborated many times, as you'll see, with Ellen Kuhn in Stanford, that has done such wonderful work on the topic also. The talk I'm going to give is based on a book I wrote on the topic, which is called The Mathematics and Mechanics of Biological Growth, but also some recent work. And you can find that if you go on my webpage. Most of it is downloadable. Don't hesitate to ask me if you need anything that you cannot access. Okay, so let's start. So first, the first distinction when you talk about growth, that if you look around you, there are essentially two main type of growth processes, either by accretion or by volumetric growth. Uh, Accretion is very typical for hard tissue, things like I'll show you seashell, but horn, teeth, bones, coral, and things like that. You have a hard material, and what you're going to do is add new layers every time during development. And if you think of the skull as also a hard material, you see that the same idea also applies to the growth of the skull. Now, volumetric growth is a little bit different, and that's typical for softer materials, soft tissue. And the idea is that if you have a tissue or an organ, and if you look at a little patch of it, think of it as a patch of cell, typically this is going to change. You know, nutrients are going to come, you're going to cell division and so on. And so if I isolate, let's say, a little sphere, what I will have through growth is, at the end of the day, a little ellipsoid. So G, what I call G, is really describe the growth of a local volume element. It's actually a linear map acting on the tangent space of the manifold, not that we care. You can think of it as a tensor. It's a better way to think of it. It's just a tensor that locally transforms a sphere into an ellipsoid. Now, the first point before we talk about the mechanisms that induce growth and how it's coupled to other fields is just a description of growth. That's why I call the kinematics of growth. And that has been done for a long time. For instance, plant physiologists in the 19th century, for instance, Muller and Sachs and other people like that, did very good experiment on the topic where they mark the points along, let's say, a stem, and they follow the evolution of this point in time and see how the points stretch. And this is really the process of volumetric growth. Each little section of the plant will actually increase, and then you can plot it, and the slope of that plot will give you the stretch at each given point. And so that'd be a characterization of the growth of the material. In terms of the G, it would be the G is one-dimensional, and then the tensor really reduced in one dimension to a single important parameter, which is how much of the material has been extended in the process. You can do the same in 2D, and again, the plant physiologists were very good. They were very quantitative at the time in that in biology and physiology in the end of the 19th century. In many ways, biology became much more qualitative after that, before it went back into a more quantitative area in the last maybe 40, 50 years or so. So in 2 dimension, you can mark point. That's with the experiment, you can mark point here and look at the evolution of leaves. And you see that all point of the leaves are actually growing. The leaf doesn't grow by adding material on the side. So this is what we call volumetric growth, in this case in 2 dimension. So once you have that, once you have a description of growth, let's say to the tensor G or something like that, you can ask question about growth. The typical biological question is to relate growth to some form of genes or genetics. For instance, going back to the example of plants, you want to know what is the effect of different type of gene if you turn on or off the gene. Is your leaf going to increase in thickness, or in length, in width, and so on? And that you can associate. So you do a series of experiments, you knock off a gene, and you see the change in the overall geometry. And by and large, this is still the main important question in biology. How do genes affect growth in biology? It's a fundamental question. But there are other problems, and problems that are more of direct interest to me than gene, is to try to develop a theory of growth based on other fields. And an important field, viewing growth more as a mechanical problem, is to relate growth through forces, for instance. And here is a beautiful experiment that people used to do in the 19th century, where you take a plant, you take a stem, and add a little string with a weight, and you mark. You make it so that you can mark the height of the stem as a function of the weight g. So in a sense, what you're doing is that you're pulling on the stem and see how fast the stem grows as you pull. And you do that for different weight. Here is a modern version, a century later, of the same type of experiment, where further weight were added. Every time 9.4 grams were added to a stem that being the leaf of corn, maize, that's being pulled. In different baths and so on, but that's not important. But what you see at the length as a function of time is that the slope increase. So if you look at the rate of growth as a function of the applied force, you see that g dot over g, that's the rate of growth, that's lambda, so the stretch as divided by the rate, the derivative of the stretch divided by itself, is a function of the applied stress. And that's quite, we understand that. I mean, we understand that for plants, of course, doing experiment like that. But you know that if you exercise more, your heart is going to change shape or thickness or things like that. You're going to build muscle. Your arteries are going to change and so on. And these are all due to mechanical stimuli at different level within the body that are exerted on the cell and force them to remodel and grow. So as a general framework, when you look at the problem of growth and over all possible scale, you realize that there are multiple scales. What the fact that growth happens depends on protein. For instance, in physiology, mostly depend on collagen and elastin for most of the soft tissue, but also fibers and cells. And at a higher level, tissues and organs. All these play an important role. The time scale involved are also very different. You have elastic time scale that essentially scale with the velocity of sound waves in the material that are extremely fast and are typically not that important for growth problem. There are viscoelastic time scale that give you a time scale, the relaxation of the stresses once you impose a stress. There are forcing time scale, or do you force the system, for instance, in your material, or how you force by a non-constant stimulus. And then there are growth time scale that takes place of the order of minutes, hours to years, or dozens of years, or hundreds of years in the case of giant trees, for instance, that are still growing. So the division of time scale will be very important for us because it will allow us to separate them and look at the problem at different scale for us. Especially the larger scale, the growth scale, we'll see directly that what happened at the elasticity level, at the elastic time frame, is not as important and can be neglected. So just for words, growth is the slow change of a body or of tissue with mass increase or decrease in the case of atrophy. We're also very much interested these days in atrophy, for instance, of the brain. Mathematically, whether you have an increase or decrease is not important. It's the same formalism, of course. Morphogenesis is typically the change in the position of tissue, and that's done typically without mass increase. It's a reorganization of the tissue that takes place mostly during development. And then you have remodeling that takes place in homeostasis during the time, the normal aging of a body, and that's really the evolution of material properties. So you might have a change of density. For instance, your bones change density with time, a change of moduli, tissue that you have, the skin does not have the same property as it changes in time because you have a constant deposition of collagen, and the type of collagen you have might change, and the way it's deposited might also change. You have a change of viscous response and all that. So a remodeling is typically an evolution of this material property. So I promise you in the title that I'll give you 10 ways to model growth. So let's start with number one. So number one is really the accretion process I started talking about that I call surface growth. So in this case, think for instance of a seashell, and the seashell right at the outside of the seashell at the rim, the idea is that the mollusk, the actual soft animal that lives inside, is going to come typically once a day and going to surround the rim and deposit small new layers of hard material. And so mathematically, you can think of it, I have a curve, and what I do is I define a velocity vector so that at a later time, I have another curve, and this process continue. So the key here is really to understand what are the laws that govern this velocity of accretion, the velocity process. So seashells are particularly beautiful to do that. And early on with Derek Moulton, we try to establish what are the typical laws that a seashell must obey in order to have certain property, for instance, to have logarithmic spiral, self-similarity, scaling invariance, and all these property. So you can find what are the conditions on the velocity vector that would enforce the existence of this beautiful regular structure. But once you have that, you can start making variation and try to understand the morphology of the patterns that you see in all these seashell, like fractal spine, sharp spine, the ribs, the wavy ridge. Actually, fractal spine is still the one that we are still struggling around. We've made a lot of progress, both with Derek and Régis Chira in France. We've worked a lot on these over the years. So the idea, for instance, so if the mantle is that the soft part of the mollusk, it comes out of the shell and they attach itself to the aperture. But if the body grows at a different rate than the deposition takes place, it might not have all the space that it needs to establish itself as a nice circle. And so what happens in a lot of these cases is that it has a wavy shape and the deposition is not strictly circular. And that's what gives rise to this beautiful shape. And let me give you a little movie out of that for different values of the parameter in the model. And so the idea is that the mantle comes and deposits, but since you have an instability at the surface, this instability takes different shape and grow differently with time. Another interesting application of the idea of surface growth is for skull growth. Skull grows mostly, which also grows a little bit in thickness, but mostly grows at the suture. You have different bone plates on your crane if you look from the top, and the suture is softer material where growth takes place. So the growth is really going to be accretive growth right at the level here of the contact of this suture with the plate. So what's interesting is that when the suture closes too early, when a baby is born normal, in a healthy case, the suture are all or still well developed and keep growing, and the skull keep growing. But in the case of craniosynostosis, a pathology, you have an early closure of this suture, and what happens then is that you have an overall deformation of the skull that can be very detrimental, can cause increase in intracranial pressure and developmental problem. So with the group of Ellen Kuhl a few years ago, we decided to develop a few models for that. I'm just showing the simulation that went with the more analytical model, where we look at accretion in different zones, and we try to see if the closure of this suture, the early closure, would reproduce all the patterns that are actually seen. So in the healthy case, you'll have growth in all along the different part of the bone, right? Accretive growth, surface growth, and you'll have a nice, well-rounded skull as you expect. But if you close one, let's say if you do not grow in this red part or in the middle part here, the coronal suture, then you'll have an expansion of the skull that is due to internal pressure and other factor. And these are well represented. And that really shows that mechanics in this case plays a very important role in determining the shape that you have in this skull. So just to conclude about my first case, is surface growth. I give for each way to model growth a little scorecard that you find at the bottom here, where I discuss it from different point of view. The computation part, at the level of the computation, it's hybrids. Typically, you can do it from a purely geometric perspective, but you have to somehow couple that to another model that explains where this deposition vector come from. From the mechanics, you can have mechanics, like in the case of the seashell. Essentially, you couple mechanics with the kinematics. You have a soft object living in a hard object, and it's the coupling between the two that is important. So at the level of the biology, the key is the rule for deposition. The actual increase is quite easy. It's just a geometric, purely mathematical problem. But the key is the modeling of this rule for deposition. At the mathematical level, there is no problem. It's well founded. We understand all the mathematical intricacies of this process. Now, for the rest of the talk, I will be focusing on volumetric growth. So remember that G described the growth of a local volume element. And one of the main distinctions of the way you approach this type of problem is to decide whether this local volume element is infinitely small. And in this case, you would shrink it to epsilon and take the limit, in which case you would define a continuous model, and you will be in the realm of field theory that you have in physics, like electromagnetism or gravitational field theory, the whole system being described by a set of nonlinear PDEs that are actually typically more complicated than the one you find in most physical theory. Or you can say that the local volume element is actually a finite size, and that leads to discrete models. And of course, many biologists really like that approach because they think of a finite element, the local volume element, as one of the units in biology, typically a cell or something like that. So that gives rise to discrete models. So let me tell you about different type of discrete model. The early on, I mentioned it because for mostly historical reason, are discrete models. They're on lattice model. And the idea, like coming from this early paper, 1985, which was called Recent Progress in Modeling. So tip to everyone here, do not entitle your paper Recent Progress, because that doesn't age too well. But the fact is that it's still a very good paper because it had a lot of good idea. And the idea is like the game of life. You have a grid, and you have normal cell and say tumor cells. And then you have rules at the interface, or the tumors or the normal cells become tumor cells. Then you can say that this part has grown because it has invaded normal cells. And you can try to do it in 3 dimensions. These early ideas didn't really work from a modeling perspective. They were good. They were easy to do, but as soon as you play this type of game, you have a lot of problem. The type of geometry that you have, for instance, a cubic geometry, you would find cubic symmetry in the system and so on. There is no mechanics whatsoever. Very little biology. After all, it's the game of light. This is just like a rule. And the mathematics, essentially there is little mathematics or no. I mean, unless you go really in the kind of game of life like Wolfram does and all that, where you can do pure math and things like that. But in terms of biology, biological model, there is very little mathematics that you can say about the evolution of this system. But these ideas led to a more refined model. They're still on lattice, so you still have a lattice, but now you're going to give a number of points on the lattice. You're going to attribute and say, for instance, that it describes the border of a cell. And these are very good idea. Now the question about changing a cell from one type to the other can be done by the use of a function. Typically, this function I call Hamiltonian because people come from physics and they like to call everything an Hamiltonian. There's very little to do except they look a little like spin model. But the idea is that you have a certain number of penalty that's going to tell you with a given probability how do you change any cell type at a given moment, and you're going to evolve the system. So you change a cell type by randomly picking a cell and sampling its neighbor with the probability that depends on age with a certain level. And so what happens is that if you want to describe growth will be contained in the term that describes the penalty in area. you change the area, that means the cell is more likely to be in a state that is larger. And doing so, you can get very interesting type of dynamic, because one of the things you can, for instance, by this type of dynamics, due to both surface tension and growth, you have you can have a cell sorting. You have dedicated computer package like CompuCell 3D that are used for this. Here's a very nice application to the formation of vertebra. You see the vertebra coming here, so it's somito formation, or in the process of leaf formation and growth. This is more than just the cellular pods model. There are a lot of variation on it. So in a lot of this discrete system, you'll have the computation is actually often extremely easy in 2D and become quite a headache in 3D, because the law change and following the geometry in 3D is more difficult. But people have done that, and it's much more common. And with the power of computer, now you can actually do a lot. There's very little mechanics. I mean, you have like a term that's supposed to represent surface tension, but there's very little you can really tell about the mechanical interaction of cell. The biology is interesting because a lot of the interaction arise at the cell membrane, but you find in this discrete system, and like many discrete type of computational approach, that there are many rules that governs everything. You have to make choices, and it's not clear where these rules come and what the effect of these rules really are on the system. There's some mathematics, but it's also limited. So it's mostly a computational approach to the problem. Now, another type of interesting model that applies to other system is a still discrete model, but they're off lattice. No, you don't prescribe a given lattice in your space. What you're going to follow is the position of the, let's say, the center of a sphere. Typically, it's sphere. Anything that's not sphere, again, is going to be problematic because you'll have to follow the geometry in 3D. But what you do is, let's say, I have the position of sphere I and sphere J in 3 dimensions. There is very nice tool like Voronoi tessellation that allows you to track these and represent that very nicely. But what you're going to say is that they interact to certain force, like it is a Newton equation, but since you are highly viscous, you can remove the inertia term. And then you obtain an equation that tells you about the viscosity of these based on the force of interaction. And the whole question now is, what is the force of interaction? And you have to decide of a certain type of potential. And that's also a bit of a problem because you have plenty of law that can derive from a potential and no, but you have to make a decision about what is the interaction. Typically, you want interaction that is attraction at long term and repulsive at short term, that express the fact that the cell cannot touch each other, right? They're crowding each other. Choosing the right one is a bit of an art, and at the place where you'd like the real physics to come in, humanological guess for that typically. But the result could be quite surprising. It's very interesting. So for instance, Dirk Drasdo has been working on this and has very nice application on this and he's worked a lot about how to define properly cell division in this system. Here is a very recent example where he looks at liver growth and tumor in liver. So you can actually do a lot about this system. But that really describes systems that mostly interact in a way that can be quantified by the position of the center. There are also computer models that do that. Chase, that's been developed for many years in Oxford, can do this type of computation easily in 2D, or even with a little bit more work in 3D. So this is done by people from computer science. So the coding is actually quite robust for this. It's a very robust program, and you can play. So the advantage is that you can change the type of interaction and see, for instance, typically you want to see a tumor is developing in different circumstances, for instance, in crypt and things like that. So one of the problems that's known is that when you look at the mechanics of these, it's also a bit problematic at some time. So here is a nice article in 2009 where they look at that. What you can have if you don't take the right force, you can have the whole system collapsing on itself. Okay, so you really have to play this game and be very aware of that. But also what you realize is that if you make a system like that of interacting things and you try to compare that to the mechanics of a system of cell and try to do simple experiment like shear, you also get in trouble and get a behavior that might not be physical. Typically, all the discrete model have problem with shear. I mean, in extension, one dimension, it always works. You can always cook the book some more to get the right force so that a one-dimensional extension works. But shear is very complicated in all systems. And so having the right physics of shear in a discrete system is not trivial at all. Again, the computation average, I would say they're trivial in 2D, but again, in 3D, that becomes a little bit more time-consuming or harder to do. There's some mechanics, but I just show you it can be problematic. But people have worked for a long time on these, and they have a good feel about the type of potential. The phenomenology has been developed in a way that matches well a lot of soft tissue. The idea, what's interesting is that the biology is very good there in terms of cell interaction and the ability to change the cell interaction based on different clones. There's some mathematics again, but again, it's mostly a computational framework, computational modeling. Now, probably the most popular of the discrete model is also of lattice. But then you put the emphasis of what you follow now is not the center of the cell, but it's really going to be the membrane of the cell represented by typically a polygon or polyhedra, right? So you really think about you cell as a finite element, and what you're going to do is really balance all the forces on that, and that works really well for systems that are really packed. Like sheet of cells and things like that. And again, the idea is that you write an energy function or a penalty function, if you want, where the position of these nodes, that is the relative position of this node, change based on the energy of the system and the interaction with the neighbor. And that works really well. I mean, you have these, again, the problem is to do proper cell division, but people have mastered that for a long time. And this vertex model actually very good in this packed cell type, really to understand cell division and the way different clones evolve through the cell, and even the geometry now in 3D, like to see in this example of vertex modeling. They really like, for epithelium sheet, this is the principal type of modeling that is found out there. There's a lot of recent and good work related to that, for like the folding of the epithelium sheet on lung and things of that nature. You can compare that different model. I think the left one is center dynamics, and this one is vertex models. It really depends on the emphasis that you have. Do you think that the interaction and the growth of a cell mostly depend on the interaction that takes place at the membrane and that the membrane balance is important, or mostly through a global average? Again, the computation average is in 2D and difficult after that. There's some mechanics, again, it's a little bit problematic, but very nice result, and one of the main tool of computational biology when it comes to development. Now, let me move to continuous model. One of the classical model for pattern formation, of course, is the Turing model, the reaction diffusion model. And one of the ideas was to extend that to growing domain. And that leads to very interesting results. So if you assume that you have C as being the concentration of a chemical, and you have a classical reaction diffusion system, it could be one equation or a system of equation. The idea now is that what happens is if my domain grows at the same time, I'm not so much modeling the growth of the domain, I'm imposing the growth of the domain, and I'm looking at how it affects the patterns. So you posit the evolution of the domain, and that change, of course, the equation by putting a convective term in the equation. And that leads people who started with this idea at Crampin and Philip Mayne, that leads to very interesting type of bifurcation that you would not see in a classical reaction diffusion model. And it is a very nice application, for instance, in the development of mouse limb and things like that. So the computation now relies on classical ID from reaction diffusion, from nonlinear parabolic equation. So you can use all the tools that have been developed in applied mathematics for that. There is strictly no mechanics in this problem. This is more only following concentration field. The biology is very interesting because that links to Turing patterns and the evolution of morphogens and things like that. And these ideas are, again, very important nowadays. And one of the big success, I think, of the entire Turing modeling and this type is that the ideas are now the basic theoretical framework for a lot of developmental work purely in biology. So even without the resource of the model, I think the most important aspect or the most important impact these theories have is that they provide a theoretical framework in which different type of experiments can be done. For instance, there's a very nice paper on the stripes of cats that was published a couple of weeks ago that doesn't do any modeling, but really use the language and the idea of these models to explain the experiments. And the mathematics of this bifurcation, of course, is very interesting if you like bifurcations and pattern formation. Okay, let's move on to number 7. So number 7 are typical of what I would call fluid models. So there is mechanics, but mostly in fluid. And the idea is the general field that's become actually huge over the last few years of active fluid, is that in certain biological system during development, the development is so fast and the interaction between the cell is so weak that the system behaves rheologically or in appearance by observation very much like a fluid. It flows with time. And so the idea is to look at cell density, include a growth and death term, and have a balance of force based on the basic equation of fluid mechanics. That means you relate the derivative of the stress to the strain and other quantities, in this case, the cell density and things like that. And the idea, including an active element, is that the way the cell interacts is to a stress dipole. And so there is a very nice body of work now looking at all the implications of these active fluids. So the computational part now can rely on all the tool of computational fluid dynamics and on the rheology and all that. The typical mechanics is based on a linear version of the elasticity plus an active version that makes this problem quite interesting from a theoretical point of view. In terms of biology, it's very good as a description of these very fluid, very quick active processes that happen during morphogenesis. Very early on during morphogenesis, where there is not quite yet a more solid component in terms of the mechanics. The stress that cells exert against one another is mostly through pressure and shear, but not through other elements of the stress. And the mathematics is also quite nice. There's a very nice body of work, both originally from the physics community and now in all applied mathematics related to this system. But if you look at more mature systems, for instance, a fully grown arteries, the type of response that you have is not at all related to this fluid system. So in a way, it's a completely different structure system that requires different tools. And one of the classical examples, for instance, that has been developed by Fung, was a great biomechanician, really one of the father of the field. And you'll find that this is the cover of his book. And one of his great discovery was that if you take an artery and slice it, you take a disc of artery and cut it, the artery would open. And what that indicates to Fung and to everybody else after that is that in the normal state, the artery contains stress and the stress are relieved when you cut that. And so what he further showed, and many people have showed since then, is that T-stress are very important from a physiological point of view. For instance, if you look at the hoop stress that is in the system, so the stress that are along the angular direction, or the hoop direction, in the absence of residual stress, the hoop stress are very large, close to the inner radius. Whereas with residual stress, they basically flatten. So these residual stress are built in the system in order to have a very flat hoop stress that prevents a lot of mechanical problem in terms of function. So it plays an important physiological function that happens during growth, that is built during the growth process. But not only that, not only residual stress is important, the type of mechanical response that you have is also very important. So if you take an artery, and you take a piece of artery, let's say you do an experiment typically out of a pig, and you put it under tension, for 0 Newton or 3 or 4, 5 Newton. Typically within the body, your arteries is being pulled by the rest of your body with around 2 or 3 Newton, around this here. And what you do is that you inflate it, and what you'll see is that the stretch, so here's the stretch and here's the how much you inflate it, so the internal pressure. The stretch between the two endpoints does not change when it is under tension, right? It's quite remarkable structure. So let me repeat that. So you have an artery, you have a tube here, and in normal physiological condition, it's being pulled by a certain tension. It's attached, let's say, to 2 points. And when the body flows and the pressure goes up and down, there is no change in extremities. So these are very different, very remarkable property. If I take a tube like that and put it in the same condition, what I will see is the 2 endpoints going up and down. But it turns out that in physiological condition, a normal artery will not oscillate back and forth as the pressure changes the radius. And you see that it should, right? Because as the pressure increases, well, the radius increases typically. And if the radius increases, the material is mostly incompressible. And so you want the length to shorten. Now, this type of behavior is not possible with this type of material, with a rubber. What you'll find is that you need nonlinear laws. If you try the naive approach that says, I take a Hookean material, rubber-like material, I obtain this type of response rather than this one. Completely different type of response from a material. So not only residual stress is important, but the type of material response that you have is also important. And that you see that in most of the soft tissue that you can think. You see that in heart, you have residual stress and the importance of nonlinearity in the constitutive law, residual stress in the heart. Again, Funk has been very happy cutting things left and right. In the esophagus, here in the trachea, in the brain, very nice experiment that really changed the way you look at the mechanics of the brain early on by the group of Phil Bailey and Larry Tabor. Even in bones, if you make a little slit in bones, you'll see a little bit of opening. It's not much residual stress, but it's there. You have that in aorta and so on. So you'll find it everywhere, even in tumor. If you slice a tumor, you'll see that opening. So during growth, it has built these stresses that you can relieve by cutting. And so the question is, how do you model that? How do you think about that? Because all these aspects have a key to understand the correct functional properties of the system. And the basic idea is the following. You can really understand it from a continuous perspective. Suppose that I take a little local volume element and all the one around also grows. Well, they're going to be very crowded, right? So the idea is that naturally, if I can isolate a local volume element, it has a certain law of growth. It was to increase to, let's say, a bigger sphere. But since all the sphere are connected, they cannot fit properly in the material. And so they start pushing against each other. And this is the stress that's being built in the system. So the basic approach, the basic mathematical framework for that is to look at, let's say, an initial system that has no stress. And then you let each piece of the material, each infinitesimal piece of the material, to grow as it wants to grow. And then you reattach all the pieces by saying that all the neighbors should still be neighbor, because you assume that there is no fracture and that the pieces are still connected to each other. And so the third step, so you first have growth, and then the elasticity, you have to compress some part and pull other part of the material. And this is what I call morphoelastic model. So it's a continuous type of model that's based on mechanics, but it's really mostly valid for material that much more solid type, for which you could actually measure a type of elasticity in the material. So these ideas were first introduced in polymer swelling back in the 40s, but it's really to the work of Rodriguez and seminal paper by Rodriguez that in '94 that these ideas were really pushed forward to as a modeling tool to describe growth. You find the same idea in many other fields, like elastoplasticity. Any form of elasticity that has a non-elastic component can be written as this type of decomposition, thinking of a change of reference configuration followed by elasticity, including fluid mechanics. Fluid mechanics, viscoelasticity, any type of anelasticity is any type of these phenomena can be written within this framework. But we're mostly here interested in morphoelasticity. So just to give you a little bit more of physical insight, suppose that now I'm placing myself completely in 2D. So I live in 2D. And what I do is that I have a ring, it's a rubber ring, and I'm going to assume that each part of the ring grows uniformly, right? So my growth tensor would be 1 in the radial direction. So it's a growth tensor written in cylindrical coordinates. So 1 means the thickness doesn't change, but each part grows with g theta, let's say, bigger than 1. So as it goes, g is like 2 or 3 or 4. So each part of the ring grows. So the g tensor, what it tells you is that I do not care about my neighbors. I grow as I want. And in this case, the material would be forced to overlap it itself, which is not possible in 2D. But you can think of a series of disjoint pieces that exist in 2D. If the material live in 2D, you'll have to glue this part to this one, and this is the ring that you would obtain. So you start with this one, the inner ring, and then you let it go, and you obtain the bigger ring. The fact that I had to force it and glue it back means that I had to put some force on the material so that it takes the shape of a bigger ring. That means that there is a residual stress in the material. And a simple example, like the Fung example, would be to cut it at one point. And if I cut it, I didn't do a great job here. I just put two pieces together. But if I cut it, it would go back to this shape, which cannot exist in 2D, right? But so the important part is that it cannot exist in 2D. So this growth tensor take my initial system in a place that cannot really exist in Euclidean geometry. And we'll see that important fact, mathematical fact, reappearing in a minute. So, but the total deformation for my configuration is the product of these two, G times A, or A times G, right? Because you first do G and then A, so you write it F equals AG. So you can do a lot with these, and let me show you an application of that. Initially, with Martin Benhamar, we thought that we could use these perspectives that was introduced earlier to try to understand certain instability that occurs in morphogenesis that are induced by growth. And here is a very nice example that you find in the brain. If you look at the brain, you'll see that the cortex, the upper layer that is rich in the cell nuclei, is typically wrinkled in most brain, most human brain. In healthy brain is highly wrinkled. That was give the appearance of a walnut to the brain. And the question is to understand that. Now, the model or the accepted model, not accepted by all, because in neuroscience, nothing is ever accepted. But from a mechanical perspective, what we think happened is that the cortex grow faster than the substrate, than the white matter that's underneath. And the two being together, there is a critical value of the cortex growth that will change the property to an instability that would lead to an oscillation. And we believe that this oscillation that is the onset of wrinkling. And much work has been done both in biology, biophysics, mathematics, engineering, and about this. It has a lot of application in material science, the wrinkling of bilayers, but also in biology. Here is what happens in a simulation. I'm increasing T and I'm increasing at the same time the uniformly the growth, and you'll see that you have a bifurcation that leads to an instability and a new pattern being formed in the upper layer. Now you can see that appearing in for around week 28, you have the formation of these early wrinkles that you can understand. And you can show it's related to pathology and things like that. That would be yet another tool, another talk related completely to the brain. And it's also true for the full two-dimensional version of the brain. And you can understand how different brain shape, for instance, the hyrax versus the wombat, has organized its pattern differently. For the hyrax, you see the patterns are elongated in that direction. And you can really understand from a theoretical point of view the orientation of these patterns by looking at the coupling of the curvature of the brain under with the onset of the pattern. So this type of brain is dolichocephalic, and this one is brachycephalic. So we had a lot of fun with Ellen and Sylvie and others playing with that, and we're still very much interested in applying these ideas to the brain. So these morphoelastic models, of course, these are my favorite. I spend a lot of time with a lot of different applications, as you can see that. You can include in them many other aspects. You can include viscoelasticity, tissue contraction, many aspects of morphogenesis, all based on the evolution of the reference configuration. Computationally, they're not easy. There are few people in the world that can really do them well, like Ellen Kuhl in Stanford. And here is an application that we did more recently on the craniectomy that is due to a cerebral edema. You have a swelling, so that can be described by growth, again by growth tensor. And the question was to find the shape of the brain as you open the skull, right, as the stress is being relieved again. So the mechanics is very nice. It's really, I think, just, I would say state of the art, because it combines large deformation, so what we call exact elasticity, with non-elastic component and elasticity that have also the proper intrinsic formulation. In terms of the biology, it has been shown to be extremely useful from the level of tissue, certainly, to the level of organs in trying to understand the shape, but also to trying to understand pathology. And the mathematics of this system is now quite well developed. So it's a very robust tool. And in the last maybe 5 to 10 years, there's been an increase in actual systematic validation and measurement of stresses and strain that allows to have much better model for this type of system. Now, of course, it has only a mechanics part, and we know from many systems that typically there are many components interact, even as a solid system, it has only one single component. But we know, for instance, for arteries that you have different components in the material, different type of protein that interact. For instance, you have elastin, collagen, and muscles. So you have an entire new set of tools that are called multi-phase model. They have different names. Let's call them multi-phase model that allow you to describe different fields located at the same place and essentially average them to homogenization in order to understand the effective load that's being borne by that. For instance, Jay Humphrey has really developed these for arteries. So, I mean, this is from 11, but he's written many papers and continue to do that. And what's remarkable in his work is that he's really done experimental work in order to find all the rates and all that. And the idea then is to not look at a single equation, but look at a series of equations and look at the average version of that. It's good for solid, but also now it's being used increasingly with a fluid component. If you have a nutrient, you can think of that as another phase in your material, and you can average that out. So for bioreactor, if you think of bioreactor when you want to simulate or you want to induce a growth of cell for organ and transplantation and things like that, you really want to combine a different type of solid that have different biological and material properties with different type of fluids. It's good for biofilms and other aspects like that. So it's also one of the main tools for continuum bioengineering, if you want, these days. So computation could be quite difficult because now you have a lot of problem in fluid structure interaction. At the mechanical, at the theoretical level, there's still some issue about fundamental properties of this system and the proper way to do the averaging that have not been worked out. There was classical work in the 90s about this type of system, but essentially there's still quite a few open questions at the theoretical level that need to be addressed to have a nice, a full theory. I don't want to use rigorous, but I well accepted where there is no gap in the system. The biology is again at the level of tissue to organs. The mathematics again needs development because you have a lot of problems related to the evolution of this system. And whereas you can write the first equation in full generality, often you have to go directly into a computational framework in order to make it, to do anything with them. There's not that much you can do and extract from an analytical point of view. Okay, so the last number 10 in terms of complexity of mathematics is what I call differential geometric models. So remember my ring here, and I say, well, we live in 2 dimensions, right? It's a fully 3 dimension. What if we could go in 4 dimensions? Where, sorry, in 2 dimensions, we go in 3 dimensions. Where 3 dimensions, I can go by taking away the glass sheet that I have, and my system evolves to this shape that relieves the stress. And this could be an unstressed configuration or very close to an unstressed configuration that lives in 3D. And so the idea is that all these stress configuration or these tissues and organs that contains a lot of residual stress could be unstressed in a different type of geometry. So the question is, what is the type of geometry in which we can formulate a theory of unstressed material? And the idea is that the reference configuration now could be viewed as a manifold with a given geometry. And the current configuration, the one that you actually observe, is a mapping from this manifold into the Euclidean world. And what we've shown over the years with Arash, we've worked a lot of that for the last 10 years, is that indeed you can, if you look in differential geometry of manifold, you can show a strict equivalence between the idea of growth and the idea of curvature and torsion. So curvature torsion, curvature tensor and torsion tensor of manifold that you can associate directly to growth. And that the correct way, if you want the most rigorous way to look at growth, is to look at a solid that is represented by a manifold with these intrinsic properties, these local intrinsic properties like curvature and torsion, that is being mapped to the Euclidean world as you study the system, as you realize it in the real world. So computation is quite hard, as you expect in this type of geometry. It's actually harder than your typical general relativity that leads to Riemannian manifold. The case of growth is actually what is called a Weizenbach manifold, the simplest case, where you have non-zero torsion but zero curvature. So computationally, it's quite difficult. The mechanics is very rigorous because now there is no approximation or hand waving that you find in other theory. The biology is possible, but I don't know a real case where it has been applied successfully to biological system. The problem is trying to relate this very high dimensional abstract notion to the biological reality. Okay, I can say, okay, how is the torsion evolve as a heart growth or artery growth or system develop? It is actually very difficult to pinpoint exactly or write a full theory that contains these 2 components. But the mathematics is absolutely superb. It's really beautiful. So that was number 10. Let me conclude by giving you a rough summary and some perspective. I show you the discrete model. They're very computationally appealing. I mean, 2D or 1D, everybody can go on the computer and do that. They can be nicely related to things like biology of the cell, cycle and claw. You should apply a cell center model if you have weak wall and a vertex model for strong wall. It really depends on what matters at the physical level in the interaction between the cell. It provides a lot of insight. Now, the mathematics and the physics of these things as mechanical object, it's much more problematic. But, you know, in a way, who cares if it works? That's often the approach seen in this type of field. When you get to the tissue level, then you get into problem. You can do it to a certain level, but you know, modeling correctly the extracellular space and tissue is not so easy. I mean, people are professional in that, so of course they do it, but I think it's not quite clean in many ways. So the discrete model are very good, and it's the tool that you want for when you study cell or collection of cells. But the continuous models are also quite interesting. Well, for one thing, you have the power of the continuum, as we know. You know, when you can write a PDE, when you can write an ODE, you really gather a lot of things in a very simple, compact way for which you can extract lots of insight. It also gives you a general conceptual framework that's based on very explicit physical principle. You can write your assumption and the laws, which is very difficult for this discrete model because so much is hidden in rules within the code, right? The number of parameters and the number of rules that you have just explodes in this high dimensional computation. There is a well-known computational framework associated with continuous model, you know, CFD and people doing finite element. The type of model that you should use is really depend on the Vertebra number, how fast that your material flow compared to its intrinsic properties. It's well established mathematically and mechanically, but often it lacks cell biology. And the real question is, how do you really do good modeling of cell biology within a continuum perspective? But at the tissue and organ level, it's very well accepted. Now, as growth is concerned, the real question is, how do we write general growth law that relate not only to the genes, but also to the other mechanical quantities like growth related to stress? So this is good at the tissue and organ level. And often the question, as you can suspect, is how the two connect, right? The typical approach, the physics approach, is that you have a bunch of atoms, you upscale them, and you do multi-scale model to get to the continuous model. In this case, it really doesn't work in this. I've never seen really a good averaging of some of these discrete models that leads to a well-understood continuous model. There's some attempt of interest, but essentially it's not the general tool. You want to be a little bit more subtle, right? You want to apply something like the Cauchy-Born rule that tells you when you have a lot of little elements, when can you actually describe them as a continuum and obtain quasi-continuum model through hybrid models, really. And this is where things are quite interesting. If you can really find a hybrid model that has the best of both worlds, that can both include the cell-cell interaction, but also the ability to look at a larger scale, then you run really something of interest. And I think all the most interesting, exciting question in growth modeling and in biomechanics related to these problems of morphogenesis is right at this level. And I thank you. I hope I didn't go too much over time. Thank you.
Thanks very much, Professor Goriely. That was absolutely fascinating and a really enjoyable kickoff to the seminar series. So thank you. Does anyone have any questions?
Not a question. Well, maybe it is a question. Have you got a review article that summarizes what you've just told us? Because it would be nice to have.
No, yeah.
It was a fantastic review.
Yeah, so I've been asked about reviews a few times, but then I decided instead to write these. And once I wrote it, I had nothing left in me to write reviews. I said it's all in there. So I don't want to repeat myself. But so the discrete model part that I have it. So the book has my interest is mostly on the continuum side, but I thought to be complete, I'll have a part on the discrete side. And that discussion that I had on all this model, there is a chapter just for that. And I really didn't want to repeat repeat myself. I said what I've said. But yeah. Happy to send you a PDF because if you want, because I don't want anybody to have to buy expensive books.
I think maybe our library has it. If not, I will make sure they do.
Please raise your hand if you have a question.
I have a question to sort of get things going. I was interested in sort of combining the ideas that you talked about with the skull growth with your brain wrinkling more fluid. And you mentioned, I mean, how changes and pathologies in skull growth can lead to differences in intracranial pressure in different areas. And I was wondering if you combined any of those ideas with the effect that it would have on the wrinkling pattern.
Yeah, so that's a fascinating question that I've been going after for many years. So the first thing you have to know, you probably know, is that the skull growth is in the skull-brain interaction, the brain dominates. That means the skull growth is completely dependent on the brain growth. It's like a glove. It's called the hand in the glove theory or idea or principle. Is that if your hand grows in the glove is going to, if it's leather, is going to give and grows with it. But the growth of the skull does not prohibit or change the brain growth. That means if the skull would grow faster, it would not make the brain grow bigger, right? It's completely dominated. That being said, now the interesting question, as you say, and we've been trying to get the data on that for many years, and it's not quite clear. You have this interesting case of craniosynostosis, where only one part create more pressure, and the question is whether or not that would change the wrinkling at all, the normal development. It's not known. It's still an open question, and I think it's a fascinating question. It's quite clear that the pressure, so what we've shown, for instance, is that a change in the pressure over a wrinkling pattern does not change the wrinkling itself. The pattern does not change. It's actually completely independent of the applied pressure. But there are also geometric constraints in the way the pattern develops. And maybe an increase in intracranial pressure, but more importantly, the fact that it actually goes against the skull might change the pattern. And so we were looking at this case with JJ here, who does pediatric neurosurgery, and he does this operation of craniectomy, a change for craniosynostosis. If there are cases that we can, from a brain imaging data, where in the same skull, for instance, the left and right, you have a different type of pattern that can be observed depending on different condition of growth, whether or not you can leave it expand or not. It's a fascinating question, and I don't know. And the next question is that if it does have an effect, how does that change the normal mental development of a child also? That's also an interesting question.
Yeah. Great, thank you. Any other questions?
So maybe a question about how do you test the validity of the models? So, for example, do you have some sort of statistics that you can do? You want to know if the model is right or wrong, and maybe you want to have some experiments and then some statistical theory as well.
Yeah, so whenever I open a review from one of my papers and I see the word validation, my heart drops. Validation. But yeah, you have a good point. So I don't know about the statistical theory. So there are different levels. I mean, the most interesting, there are people have been measuring stuff and it changed, the mass and length of animals and all that, or trees forever. But the most interesting things is at the mechanical level is when it is volumetric growth. Probably one of the most interesting part in terms of validation is the work again of Alain Goriely, was that this beautiful experiment, for instance, in skin, where what you do is exactly the same that you do on the leaf that I show you. You put markers on the skin, and then you put an expander. This is the kind of operation that's done in plastic surgery. And because you apply a mechanical load that you fully control, a geometric constraint, you expand the skin by under, you stretch the material, and the material remodel itself by growth. And then following this point, you can really pin down this growth tensor and find different part of the growth elasticity part. And you can build a computational framework that allows you to know what are the stresses related and what will be the effect of a given growth type. And these are very nice, I think, in terms of trying really to measure this relation and finding the underlying laws that govern the relationship between growths and mechanical stresses.
Thank you.
Sasha Sherman had a question. She asked, since the wrinkling is caused by an instability, do you have to take into account? Sorry, did my sound get cut off there a little bit?
Yeah, I said, do you have to take into account? And I don't know.
I'm sorry. Do you have to take into account stochasticity when modeling to get an exact wrinkling pattern?
So no, you don't have to. These are extremely robust. These patterns, these early wrinkling patterns, are extremely robust. Now, the point is not to make a prediction. We're not quite there yet. I mean, there's this good work. What you have to know in terms of wrinkling on the brain, or the way these gyri and sulci establish themselves during growth, is that there are two types. There's the primary gyri and sulci that you more or less recover in most brain of a given species. And then on top of that, you have this secondary one that are really much more like fingerprints, that they really depend on the details of the. So if you want to capture these, then yes, you would have to put some stochasticity and do some kind of ensemble average to understand secondary pattern. Now, there are different groups in the world that have been working along these ideas, and I think we close to the point where we understand the formation of the primary patterns based on differential development at different time in brain development and the underlying geometry of the substrate, which is the white matter. We're not quite there yet in understanding the secondary pattern. We understand some feature of that, but we're not there. And in that part, I think this is where stochasticity would be important. But just like fingerprints, because these are the type of instability that can occur with a very flat landscape in terms of wave number, you have a lot of possible choice, and so you'll never be able to predict any patterns, or explain any given one. What you'll be able to understand is the statistics of these patterns and how they emerge. It's a good question.
Thank you. Does anyone else have any more questions? Okay. Well, I mean, perhaps we can end here. I have put the website for the whole program in the chat for anyone who has not had that. There's also a spot on the website to sign up if you want to get email notifications of our next events and things like that. The seminar list is constantly growing, and we're working on settling some dates for those who've agreed to speak, and we'll be scheduling those soon. So we'll be sure to let everyone know when something when our next event is. So just thanks again, Professor Goriely, for taking the time to speak with us, and to everyone else for signing in. And yeah, wherever you are in the world, I hope the rest of the day or evening goes well. Thank you very much.
Thank you. Great afternoon.
Thank
Bye.
you very much. Bye.